thank you to the organizers for the opportunity to present this talk. My name is Cai Roberts. I'm an Assistant Professor of Pharmacology at Midwestern University in Downers Grove, Illinois. And the title of today's talk is Epithelial-Mesenchymal Transition in the Crosshairs, Novel Treatments to Kill Drug-Resistant Metastatic Cells in Cancer. A little overview. I'll start with an introduction on what epithelial-mesenchymal transition, or EMT for short, actually is, and how it relates to some clinical challenges across a number of different cancer types. My training was in the field of ovarian cancer, so I'll walk through some of the foundational work from my PhD and postdoc and early years here at Midwestern University as an independent researcher in the ovarian cancer field, and how that kind of set the stage for the later part of the talk. But we'll talk about our ongoing studies and some of the techniques that we're bringing to bear on the problem of drug resistance and the need for new therapeutic strategies. And I'll close with some of our future directions. So by way of introduction, in my lab, we focus on three distinct cancer types that have one particular thing in common, and that's they are a challenge to treat in the clinic. These are epithelial ovarian cancer, which I will abbreviate as EOC, colorectal cancer, which I will abbreviate as CRC, and oral squamous cell carcinoma, or OSCC. And in each of these, metastasis is a large problem that leads to much of the mortality that is observed. And there are additional problems with acquired therapeutic resistance, especially in the recurrent disease states, and poor prognosis when these are found at advanced stages. This is particularly an issue in EOC, where the majority of these cancers are actually discovered when they are already at stage 3 and stage 4. Have already metastasized and are therefore quite difficult to access all of the various tumor sites throughout the patient's body. And a process that kind of links these drug-resistant, recurrent metastasis phenotypes is EMT. So EMT is a developmental process. So this is a process by which epithelial cells, seen on the left-hand side of this figure, that are firmly attached to their basement membrane and to each other through various adhesion proteins, are able to downregulate expression of these proteins and upregulate others while remodeling their cytoskeleton and undergoing various other changes that allows them to depart from their original location and to migrate to distant sites. So this is important during the developmental process as the different germ layers start to develop and as the tissue architecture is taking shape following the blastocyst stage. And this is reactivated in cancers due to its ability to allow cells to metastasize. So a couple of these key factors from the developmental process, such as twist and TGF-beta, are going to recur as being important factors for cancer progression as well. This is taken from a paper I wrote as a PhD student, focused on a particular EMT transcription factor, TWIST1, which was shown again to be important for development. The same kind of tissue reorganizing and cell migration are important in processes of wound healing and angiogenesis, but by and large, this is limited to these few normal physiological processes, and otherwise, these EMT factors, such as TWIST1, are largely transcriptionally silenced. However, in cancers, when they're reactivated, they can lead to a number of these other processes. So TWIST1 in particular, through my own work and the work of many other groups, has been tied to cancer stem cell-like phenotypes, drug resistance, the induction of this global EMT program, and metastasis and angiogenesis of those cancers. But TWIST1 is not unique in this. There are a number of transcription factors and other signaling molecules required and involved in the EMT process, and each of them can be linked to all of these phenotypes as well, making EMT an attractive therapeutic target for future cancer therapies. And sort of to summarize a very, very large body of literature, the conventional wisdom is basically that EMT is correlated with drug resistance and therapeutic resistance overall across a number of cancers. And this can include radio resistance as well as chemo resistance. But some of previous work that I and my colleagues and collaborators have done have shown that TWIST1, again, one of these EMT markers, is responsible for cisplatin resistance in ovarian cancer. And we did some studies regarding the mechanism of that resistance. And we had a number of studies several years ago on the development of siRNA and nanoparticle delivery platforms targeting TWIST1 in order to reverse drug resistance and try and bring some of these more aggressive tumor types in check. And that included ovarian cancer, melanoma, and some forms of triple-negative breast cancer. A paper that was published by my postdoctoral colleagues in 2020 analyzed, amongst other things, TWIST1 as a prognostic marker, in which we showed that TWIST1 high and CBX7, a polycomb protein, being low, was correlated with a far worse prognosis amongst ovarian cancer patients. And what we discovered in the course of all of these studies is that ovarian cancer, this EOC, is a little bit of a special case. The profile that we saw as kind of EMT progresses over time in terms of how these cells react to drugs starts down here in the bottom left corner with our stem cell population. And unlike most other cancers where typically EMT and the more mesenchymal phenotype is associated with stem cell status, my colleagues actually found that the stem cell population in these ovarian tumors can be epithelial. And so you're at the very left end of this EMT spectrum here on the epithelial side. And as stem cells, these are more slower growing than some of your other tumor cells and are inherently treatment resistant, partially on that basis and due to their sort of stem nature overall. What we found was as these cells start to differentiate, they undergo an early EMT process, they start dividing faster. And as fast dividing cells, they're more easily targeted by some of these conventional chemotherapies that are designed to inhibit proliferation in some of these rapidly dividing cells. However, given our work on TWIST1 and CBX7 that I alluded to earlier, our work on TWIST as a marker of drug resistance, it seems that this is sort of a bimodal pattern, where as EMT continues to progress, as treatment starts to exert selective pressure on these tumor cells, the continued EMT and continued mutation of the tumors leads to a sort of secondary drug-resistant population that is, again, more mesenchymal still, but results in a drop in drug efficacy. Let's do that a little bit again, because I can't say the word efficacy. Starting from the top of this slide then. So this is a model that we have developed over the last few years in epithelial ovarian cancer, is with respect to its pattern of drug response as EMT progresses. So unlike most tumors in which EMT and the mesenchymal phenotype tends to correlate with your more stem-like and drug-resistant population, what we found in the case of EOC is that epithelial cells are actually functioning as the stem cell component. And so these stem cells are epithelial, they are growing more slowly, they are inherently resistant, partially due to the fact that they are growing at a more slow rate. But as they start to differentiate, as they're undergoing this early EMT, they start to divide faster and become actually more vulnerable to therapy. And so we start to see that drug efficacy actually increases in these early stages of EMT. However, integrating some of the other studies that I mentioned previously, it's clear that continued EMT after the selection by therapy, selection by time, and the acquisition of further mutations in the tumor leads to this sort of further progressed, more mesenchymal phenotype, which is correlated with a return of drug resistance. So what we see in most carcinomas is basically the second half of this plot, where as EMT progresses, you're getting more chemoresistance. Whereas on the left side of this plot, this is mostly unique to the case of EOC, and therein seems to lie the difference. Another aspect of EOC that we wanted to analyze more closely is the involvement of DNA repair. And about 15% of EOC tumors carry mutations in the genes BRCA1 or 2. So these are primarily known, as their name implies, as breast cancer susceptibility genes. However, they have also been implicated in, again, about 15% of these ovarian tumors, and in some other cancers as well, including, in rarer cases, some prostate cancers. However, BRCA1 or 2 loss results in a defect in the DNA repair pathway referred to as homologous recombination. And so if you have a single strand break in the DNA, as seen on the left-hand side of this flowchart, then an enzyme referred to as PARP, which is involved in the base excision repair pathway, can seal that lesion and repair the DNA. If for some reason that doesn't happen, that single-strand break upon replication of DNA is going to turn into a double-strand break. And again, at that point, there are two options. This is a bit of an oversimplification, but for our purposes, two options. One is conservative repair, referred to as HR, homologous recombination, where a sister chromatid or a homologous chromosome can be used as a template for repair, and that gap can be filled and sealed with no loss of genetic information. On the other hand, non-homologous end joining, or NHEJ for short, is a more error-prone process where the ends of the break are simply joined back together as quickly as possible, and this often leads to insertion and deletion events that may alter the genetic information at that site. And so a strategy for treating the tumors that have these BRCA1 or 2 mutations or deletions is to use a synthetic lethality approach. And so in these cells, the homologous recombination pathway is deficient because of the loss of BRCA1 or BRCA2. And so any double-strand breaks are going to be repaired via NHEJ. But the single-strand break repair pathway is still available. However, due to synthetic lethality, if we then use an inhibitor of PARP, such as the drug olaparib, to block base excision repair, these cells must now rely solely on this error-prone repair, leading to an increased burden of DNA damage and ultimately cell death. The catch with this is that only 15% of these tumors are BRCA deficient. Most of these, about 85%, then are going to have wild-type BRCA1 and BRCA2. And in many cases, we see resistance manifest through a second mutation that restores expression of the wild-type BRCA proteins, and thus they are able to undergo homologous recombination once more. Furthermore, for BRCA wild-type patients in clinic, this is still sometimes used as a strategy. However, olaparib is only used in platinum-sensitive patients as a maintenance therapy, and the overall survival benefits are as yet unclear. And a question that was asked in my postdoctoral laboratory that I have continued looking at in my own lab is, can we either overcome this resistance mechanism with the restoration of wild-type BRCA, or can we expand the cohort of tumors into the BRCA wild-type domain so that we can use this olaparib synthetic lethality strategy for more tumors than we currently do, especially in cases where tumors are resistant to the other first-line therapies, such as platinum in the case of EOC. And so if we look at our homologous recombination pathway following the break, there are a number of proteins that are required in a specific sequence in order to effect repair, including RAD51, RAD52, and BRCA1 and BRCA2. And so my colleagues and I in my postdoctoral lab used a model of epithelial ovarian cancer, two lines PEO1 and PEO4. PEO1 was derived from a patient who had a BRCA-deficient tumor who was initially sensitive to olaparib therapy. And we see if we culture these cells and look at their protein expression that they have a deficiency here in the BRCA2, and also in this left-hand column, the expression of snail, slug, and fibronectin, which are common mesenchymal markers. And the expression of ECAD-herin here is low, and that is an epithelial marker. And this is compared to PEO4, which was a cell line derived from the same patient, but once they had presented with a recurrence, this was no longer seen to be responsive to olaparib. And this makes sense because, as we can see in that right-hand column, they have restored expression of BRCA2, and thus now these cells are able to undergo homologous recombination and thus are resistant to the olaparib. Interestingly, their expression of snail, slug, and fibronectin, these mesenchymal markers, is lower, and the ECAD-herin levels are higher. And this suggests that perhaps this was one of these more epithelial-like, stem-like populations that had acquired this secondary mutation and was able to reconstitute a new tumor when occurrence manifested itself. And I mentioned earlier that TGF-beta was one of these factors that was responsible for the induction of the EMT process. And what we see is if we take these PEO4 cells that are expressing BRCA2, that are more epithelial-like, if we treat with TGF-beta, we actually see a reduction as shown by these red arrows here, in the expression of some of this homologous recombination machinery, particularly BRCA2, BRCA1, and to a lesser degree, RAD51. And as you might expect, this also translates to a change in olaparib efficacy between these lines. So what we can see in the green line on this graph is the PEO1 cells. These are these BRCA2 deficient cells, and we can see a very nice dose response to olaparib as the synthetic lethality approach is still in play. In contrast to this, the blue curve there is PEO4. This is that BRCA reversion. And so now these cells are much more resistant to olaparib, given that they are once again able to undergo this homologous recombination. Interestingly, however, if we treat PEO4 with TGF-beta, again, to start this EMT-like effect, we get this red curve where we see dramatic sensitization to olaparib, and a response is very similar to what we see in the more mesenchymal-like PEO1 cells. I then brought in a cell line model from a previous lab, R182 and Tara. These are two ovarian cancer cell lines, again, derived from a single patient sample. In this case, R182 is derived from the patient and is one of these very epithelial and stem-like cells. It expresses CD44 and MyD88 as stem cell markers, and a number of epithelial markers as well, and notably is negative here for the mesenchymal marker TWIST1. Tara is a derivative of R182 that was generated through growing R182 in stressful conditions in vitro until they underwent an in vitro EMT event. We can see this on the basis of TWIST1 here as one of our canonical EMT drivers and EMT markers. And interestingly, if we look at the HR machinery, we see the same thing that we observed in PEO1 and PEO4. That is that in the mesenchymal line Tara, the expression levels of BRCA2 and RAD51 have been markedly decreased. And similar to what we saw in terms of olaparib response with PEO1 and PEO4, we find the same thing here, where these mesenchymal Tara cells shown in the red are showing a robust dose response to olaparib, whereas the epithelial R182 cells are largely just shrugging this off. And so to summarize kind of all of this foundational work thus far, we published some of this findings regarding olaparib and how it relates to EMT last year. And this is the same graph we saw earlier, where chemo seems to have this bimodal distribution, where in EOC anyway, cells at the beginning of the EMT process are resistant, become sensitive, and then once again acquire resistance. And we see that response to these DNA repair drugs, such as olaparib, are following a similar kind of trend, although whether this trend continues upwards or shows the same kind of resistance remains to be seen and will be the subject of some of our ongoing studies. So on the subject of ongoing studies, I want to transition at this point to talking about some of the work we're currently doing and looking forward to doing in the near future. The first of these is continuing to look at these DNA repair pathways and drugs associated with those DNA repair mechanisms and associated pathways as drug targets. And also looking at drugs that are derived from some natural sources, in particular some marine pharmacognosy. And these first two angles are basically innovative in that we're looking at a collection of drugs that are perhaps underutilized and understudied in the context of these cancers. And finally, I want to go over some techniques that we have for integrating some dynamic measures of cell behavior with our drug screening and biochemical assays to kind of get a full picture of how the drugs that we are testing are impacting not only cell survival, but the EMT process and their migratory ability, kind of all in one big package. So for our first section of this, focusing again on these DNA repair pathways and some related agents. So in addition to looking at cisplatin, this traditional chemotherapy, we had mentioned that these mesenchymal Tara cells, shown in red, are part of that early EMT process that's leading to sensitization due to their rapid growth rate. Olaparib, we have also mentioned as well, where this drug seems to be more effective in these Tara cells, where they're losing the expression of some of those homologous recombination factors. We also looked at a few other drugs. First off is COO55933, or COO for short, which is an inhibitor of the ATM kinase, which is really sort of an upstream signal or a master alarm, if you like, in cases of DNA damage is upstream of the decision of which DNA repair pathway to use. And by and large, responses to this drug have been minimal across both of these cell lines. However, new 7441, which is an inhibitor of DNA polynucleotide kinase, is again showing this differential effect where it seems to be more effective in these mesenchymal cells as compared to their epithelial forebears. And DNA PK is required in the non-homologous end joining pathway, but not in homologous recombination. And so we were curious to see if we could explain why this was. And we hypothesized that, well, we already know that in these Tara cells that we're getting a downregulation of the homologous recombination pathway. And so if these cells are confronted with a double-strand break, maybe they're going to upregulate non-homologous end joining as a compensatory mechanism. And indeed, that's what we see if we look at some of these canonical pathways that or factors that are being impacted by this new 7441. We see that the Tara cells have increased levels of Q70 and 80 and activity in DNA PK, suggesting that they are relying more heavily on non-homologous end joining, and thus new 7441 is undermining that. And some future studies on this are in the planning stages. An additional drug that we were very interested in is cediranib. And as you can see in the graph here, this is far more effective in our mesenchymal cells compared to the epithelial ones. And we are curious as to why. Because cediranib was initially developed as an inhibitor of the VEGF receptor, and it is currently in clinical trials, including in combination with chemotherapy and olaparib for ovarian cancer as an anti-angiogenic agent. So this is intended to block the formation of new vasculature and essentially starve the tumor as its mechanism of action. However, we're doing all of this in vitro. Obviously, there is no vasculature being developed in our multi-well plates. So something else had to be going on. And so we wanted to know, how is this drug killing our mesenchymal cells? There was some previous work by a group at Yale that suggested that cediranib could further alter the expression of DNA repair factors, including HR proteins. And also, at the end of the day, VEGF is a growth factor. So perhaps there was some survival signaling in these cells that was being affected by the presence of cediranib. And this is work that was done by Andrea St. Laurent, who is a medical student in the lab. And she found a very similar dose response in the Tara cells using cediranib to what we had observed earlier for olaparib. And in looking at homologous recombination and non-homologous end joining factors, there were a few minor differences, including a lack of upregulation of BRCA2, as shown by the arrow here in the Tara cells as compared to R182 with cediranib treatment. But by far the biggest change that we observed came when we were looking at some of this downstream signaling from the VEGF receptor itself. So in this schematic on the right-hand side, I'll draw your attention to the area labeled proliferation, where downstream from things like focal adhesion kinase, you're getting activation of MEK and ERK, and this is leading to pro-proliferative signaling in these cells. And in the western to the left, we see that in the R182, these more cediranib-resistant epithelial cells, that the levels of ERK1 and 2 activity they're not really changing over the course of 24 hours of treatment. Whereas in the mesenchymal Tara cells, we see a very striking difference where the levels of phospho, i.e. active ERK1 and 2, are markedly lower after 24 hours of cediranib treatment, and that's despite upregulation of the total protein, perhaps in attempts to compensate. And so it seems that there may well be some of this survival signaling in the cells that are being inhibited by cediranib, and studies are ongoing at the moment in our lab looking up and downstream from this to elucidate a little bit more of this pathway. But we've also tried to extend beyond ovarian cancer into some other cancers. And the first one we really looked at was colorectal cancer, again, abbreviated here, CRC. And we have a similar kind of cell line model setup to what we have in ovarian cancer, where we have a pair of cell lines, SW480 and 620, which were derived from a primary tumor and a metastatic site, respectively, again, from a single patient. We also have 620F, which is a line that we have generated by growing SW620 in non-attachment conditions and then reattaching them again into cell culture plastic. And what we see is, if we compare, again, this primary to metastatic cell line, we see a similar pattern of expression of DNA repair factors to what was observed in the ovarian cancer cells, where we see a loss of BRCA2 and RAD51 expression in the metastatic cells, and an increase in the expression of NHEJ factors on the right-hand side, particularly in the 620F cells that have been floated and reattached to the dish. So this is very reassuring that we're seeing a similar pattern here in this additional tumor type. And at this point, I want to introduce one of our gizmos that we have in the lab. I'll get into this a little bit more at the end when we talk about kind of our integrating all of these various kinds of assays into a big picture. But we have a live cell imaging system. This is called the Cell Logger Mini Plus by Curiosis. This is an in-incubator microscope. So it lives in the incubator, maintaining a 37-degree carbon dioxide atmosphere, as you would normally have for cell culture. And it is able to monitor the conditions of cells in real time throughout the course of a treatment experiment. So it takes bright field images of the cells at regular intervals, as shown in this middle image, and then is able to analyze that image to determine the level of confluence in that well, and thus use the confluence of the well as its measure of cellular proliferation over time. It's also able to image fluorescence, enabling us to use some dyes such as the Cytox green dye shown on the right-hand side here as markers of cell death. And by using this system, we're able to monitor whether cells are simply never growing or are growing and then dying off by their growth patterns, their confluence patterns over time, and whether or not these cell death markers are being activated in these cells. And so this is an example of the kind of data that we can generate from a single experiment here, but again, we can do this for multiple runs. And if we're comparing, again, the SW480, this is this primary cell that is here in the teal color, to the purple, which is the SW620, that is the metastatic cells. The darker colors here are the non-treated condition, and we see that their growth rate is fairly similar. In the epithelial cells, the pale teal line is treated with cediranib over the course of 3 days, and we see that the growth of these cells is certainly inhibited. But if we look at the pale purple line, which is the 620 cells treated with cediranib for that same 3-day window, what we see is these cells never really start growing. And in fact, we see a slight decrease in the cellular confluence over time as these cells are starting to die off. And so this really does give us a lot more information than what you would get from your typical single time point endpoint assay, by allowing us to observe kind of these cells in real time and what their growth patterns are. And beyond this, we also, because we have all of these images and are able to collect some of these time-lapse videos of this, you're able to observe changes in morphology, which starts to inform any response that the cells have in terms of apoptosis, necrosis, or any adaptations that they may be going under, such as upregulating EMT in response to whatever treatment you happen to be using for them. So a corollary to all of this is based in some literature that showed that inhibition of PARP with some various PARP inhibitors was actually impacting VEGF signaling. And so we've seen that in the course of cediranib treatment that we're getting impacts on cell survival and possibly in DNA repair by using VEGF inhibitors. Is the opposite also true? Can we see any changes in this VEGF and survival signaling by using olaparib, i.e. is olaparib working on these cells in a way that is independent of its role as a PARP inhibitor? And so some evidence from various cell types in the literature has supported this. Duriali Mirzat, who is another medical student in the lab, did some additional pilot studies with this, suggesting that these Tara cells actually have a lower expression of VEGF receptor overall, although there is some there that is able to be activated at some of these later time points when treated with olaparib. So this is some very early studies, but still another sort of angle into this same overall sphere of drug treatment strategies. I'm going to pause for a drink for a moment, so if we can edit this little pause out, that would be great. So the second area that I want to discuss is on some marine natural products, and in particular on a class of compounds referred to as manzamines. These are extracts of a marine sponge So these compounds were isolated from sponges in the family Haliclona and a number of others, initially almost 40 years ago in 1986. And in that time, they have found use, at least in basic science studies, for a number of different indications, including as anti-malarial drugs, other antimicrobial uses, as anti-inflammatory agents. But surprisingly, given the fact that this is over a 40-year time span, there has been remarkably little work done on if these compounds may be active in cancer. There was some early study here just in leukemia shortly after their discovery in 1986. But there was a long kind of lull time until 2011 where there was some work in pancreatic cancer. There was a paper on colorectal cancer in 2018 and a couple on cervical cancer in recent years. But I want to focus mostly on this 2018 study in colorectal cancer. This is a study by Lin et al. That was published in the journal Marine Drugs. And they were looking particularly at manzamine A, which is kind of the flagship molecule of this class of compounds. And they were looking at in a model of colorectal cancer. They chose HCT116 as their cell line of choice. And they were showing that they were getting some IC50s in these cells on the order of 5 to 10 micromolar. They were getting some robust inhibition of growth in some colony formation assays. And interestingly to us, they were also seeing that treatment with manzamine A seemed to be downregulating some of these EMT genes, including upregulating E-cadherin and downregulating snail and twist. So we were curious if we would see some of these same effects in our own cell lines, and these studies are ongoing, but we have some very encouraging preliminary data here showing that in our ovarian cancer cells, on the left are trusty R182 and Tara cells, we're seeing some very robust cell killing by manzamine A with IC50s on the order of about 2 micromolar. And likewise in both the colorectal cancer and then in this green line on the right is Detroit 562, which is an oral squamous cell carcinoma line. We'll talk a little bit more about those cells shortly. But in any case, all of these cell lines are showing a very nice toxic effect with a similar IC50 level on the order of about 2.5 micromolar. And future studies on these compounds will involve looking at their ability to inhibit EMT across all of these different cell line models that we have, and also whether additional manzamines besides manzamine A may have some more favorable kind of pharmacological properties that would allow us to develop them into effective new drugs for these kinds of invasive cancers. And for the last part here, I want to talk about how we can integrate all of these various aspects together, the EMT, the drug response. And this is a project we have ongoing in oral squamous cell carcinoma. There's not really any good biomarkers for staging and use in prognostics in OSCC. And there hasn't been very much work done on drug responses across the different tumor stages, partially due to the fact that early stage OSCC is largely treated with either surgery or radiation. And so chemo is largely reserved for some of these later stage disease instances already. And so we wanted to know if we could find markers that would predict tumor aggressiveness, would correlate with EMT, but then could also allow us to predict their response to various treatments. And to do this, we have a panel of 4 cell lines. We have PGK, which are your primary gingival keratinocytes, i.e. these are normal cells. We have dysplastic oral keratinocytes, or DOK. This is sort of an early dysplasia or a precancerous lesion. We have SCC25, abbreviated SCC here, which is a locally invasive carcinoma. And then finally, we have Detroit 562, abbreviated Detroit, which are derived from a distant metastases. So this was taken from a patient with pharyngeal carcinoma, but the actual cells themselves were harvested from a pleural effusion. And what we see is some selected biomarkers that we are looking at in the lab, stratifin, GSTP, and TPM1. Each exhibit a unique pattern of expression as you go from your normal cells through your dysplasia, carcinoma, and metastatic cells. And this correlates with some of our EMT markers. So as you might expect, E-cadherin is being decreased as these tumors are progressing, and N-cadherin, a marker of mesenchymal cells, is increasing. Surprisingly, in contrast to what we saw in ovarian cancer, we see that fibronectin is actually expressed quite highly in these normal cells, and the expression actually falls off quite significantly as these cells start to progress. And so that is something that perhaps will be unique to these OSCC models. But in conjunction with all of these biomarkers, we're also looking both at drug response and cellular behavior. So we're using, in this case, docetaxel, which is a common drug used for OSCC. And we see that these Detroit cells, that are these more aggressive metastatic ones, appear to be more resistant to docetaxel across multiple log range of doses as compared to the more locally invasive SCC25 cells. And we can also correlate this with their ability to migrate and invade, again, kind of a hallmark of these EMT cells. And I'll play in a moment here a video. This is of a scratch wound assay. This is Detroit 562, just as an example, these sort of most aggressive, most metastatic cells. And if you keep an eye here as the video plays on sort of the leading edge of these cells as they start to migrate towards the center of this void in the middle, you'll actually be able to see sort of the extensions of the cytoplasm as these cells are reaching out and pulling themselves along as they begin to crawl. I hope you could appreciate there seeing those cellular projections coming out as these cells were nearing one another. And another thing to note, oftentimes you'll see in the literature a common time frame for wound healing assays. You're getting complete healing in 36 to 48 hours. Many papers are going to show you sort of the 0 and 24-hour time point with a large amount of space still remaining. This video that you just saw was a time lapse over the course of 24 hours, and you can see that in less time even than that, this gap has been completely sealed. And so that combined with those cellular projections, I think, speaks to just how aggressive and migratory these cells are able to be. So with that, I just want to wrap up with some of our future directions. Obviously, it's neat that we've been able to see a number of these drugs working in unexpected ways, but ultimately, of course, we'd like to know why that is. And so one aspect of our future work will be determining some of these molecular pathways and signaling networks that are connecting these various aspects of tumor progression together. This EMT process, changes in the DNA damage response, altered VEGF signaling, and some of these cell survival signals, and seeing how they all work together. And we've done some RNA sequencing analysis across some of our lines that we're still analyzing and will be including in some of our future publications. We also want to further explore the natural product space, again, looking at some of these other manzamines and similar marine-derived compounds. There's been a great deal of work in the use of the harmala alkaloid harmine and its derivatives in trying to target TWIST and other EMT genes. And so comparing the results that we're getting with some of the studies that have been done in that realm. And ultimately, in the long run, doing some translational studies of some of these drugs and trying to find new strategies and new combinations of these different agents that can be effective for some of these most drug resistant and the most difficult to treat cancers across a number of tumors that we get in the clinic. All right, I just want to close with some acknowledgements. The folks in my lab, Jahada Boris, who is a research specialist, who is spearheading our manzamine project, students Andrea St. Laurent and Duriali Mirjat, who were former members of the lab who I introduced earlier, Josephine Sang, who was, you saw on the slides, who was responsible for our SW480 and 620 study, and Mohammed Hamoui, who is a master's student in the lab, who is in charge of our OSCC project. I also want to acknowledge my collaborators here at Midwestern University, Drs. Marcia Pierce and Alejandro Mayer, who are working with me on the Marine Natural Products project, and Hilal Arnouk, who is an expert on OSCC and is helping us out with the oral squamous cell project. The heads and members of the Ratner Lab at Yale University and the Moore Lab at Wayne State University, my postdoctoral colleagues, Midwestern University's Department of Pharmacology, and our core facility for all of their resources, and of course, the funding support for this work. Thank you for joining me for this talk. I hope it's been informative, and I hope maybe to collaborate with some of you in the future if this work has been of interest to you. Thank you.